So we're talking today about the DNA replication fork, although that has changed a little bit with the recent development in the scientific community regarding how the two strands are actually oriented around the helicase. And that, of course, is something we're really proud of in New York State because that was actually discovered and written about at the Brookhaven National Lab here on Long Island. That's where we're from, so we're proud about that. But today we're going to get into the nitty-gritty of this, and we're going to look at these two nucleotides, um, guanine and cytosine here, and we're going to look and count these carbons because we have to know something about the direction of how these two anti-parallel strands are built in our double helix. So this is the first carbon, and of course, for those not knowing, this five-sided pentagon here is really the deoxyribose sugar, and we count by the carbons starting one. This would be the second carbon, and of course that carbon would be right at this corner here. So this is the first carbon. Right here is another carbon, and we do this in organic chemistry. We like to things that make shapes. We look at the carbon backbone, so we don't write them. So this is a carbon here. That's the first carbon. This carbon here attached to this hydrogen and oxygen here of the hydroxyl group is the third carbon. That's a really important one. This is the fourth, and this is the fifth. Let me go in this anti-parallel strain. We are looking at, if you can see that still, the first carbon is always the one attached to the nucleotide here, and this would be the second, the third, the fourth, and the fifth. Now it's really important that you understand that in this strand, okay, we have, this is the 5' ending, and this is the 3'. So we're going from 5 to 3 in this strand. And in this case, all right, we have the 5' ending, and we have the 3'. So this strand is going 3 to 5. Now, it's very important that we understand that DNA polymerase 3, okay, is how we actually build these. Now, we often study these in prokaryotes, and in eukaryotes, they're a little more complicated, but the same process does occur essentially. So we can actually study one and make comparisons. So no problem here. But what we're going to talk about today is when we start looking at the replication fork, and I guess that fork is going to change again because of the scientific changes I just mentioned, but that's going to change a little bit. But when we're talking about the replication fork, we're going to talk about which direction the building of the nucleotide goes. So DNA polymerase 3 attaches another nucleotide right at this carbon, although my arrow seemed to stop there. But basically, we want to attach to this hydroxyl group that's on the third carbon. So DNA's polymerase can only add to the 3. It's going to get complicated with the 3 and 5s, and people will tell you different things. I want to tell you something very easy to remember in all of this. We always add to the third carbon, or the 3' carbon. If you just remember that, and that's how you build this replication fork, and the things that you're studying in your classes will be much easier to understand. So we're always going to build to this carbon here. Now, if you look on this other strand that's going the opposite way, where's the third carbon? Okay, it's right here. So we would build in this direction. So as I said before, we're going to add another nucleotide in this direction, or at least DNA polymerase will help us do that. And of course, we would also build in this direction. So as you can see, the reason why we can only build in these certain directions is DNA polymerase 3. That enzyme is essentially a three-dimensional structure that can only work in a certain direction. It only kind of holds a nucleotide and able in its three-dimensional shape attach this phosphate group, okay, I'm sorry, to attach this to this oxygen here. So because of this phosphate group, or this oxygen here, another phosphate group, and because of the three-dimensional nature of the enzyme, it can only do that in one direction, okay, unlike the band, which I don't know if is any good. All right, so any case, so we're building from the 3. It's important. And we're either going this way or we're building in the other way. We can't build from these positions here. So we always add from the third carbon. And that should make some sense here as we get into the diagram that I'm going to draw below. That's very famous. Now I'm going to draw a little opposite of probably what you see in the books, only because I want to make sure that you can have a new way of looking at it so you can identify it. So what we're going to have is our double helix here. Okay, looks like some kind of sine waves going here. Right, something like that. And of course, it's attached. These are the nucleotides here. And what we're going to put is our helicase, which is the enzyme. We'll put an H here. That is basically unwinding and unzipping the two strands. And let's just mark this strand here. So this top strand, let's make that the 5'. Let's make this the 3' ending here. And if you notice, if you follow that through, this would have to be the 5 ending here. Okay, but I'm gonna make that longer. So this would be the 5' ending. And of course, you can visualize what that means. The 3' ending means we're ending with what? We're ending with this side of the nucleotide, this third carbon. 5' ending means we're ending with this sticking out. Okay, and because it's anti-parallel, building in different directions or moving in different directions, that could be expected. And again, if you can visualize those things, it makes life easier. They're not just numbers. So let's get back to reality. Up there goes gravity, and here we go. So let's continue our strand. So this strand will just continue up. I know I put a break there. And in fact, let me try to write over that. So continue on, and it's unzipping. And let's take this one, and it's unzipping too over here. And we can add, and I'm just showing the basic nucleotides popping out here. No big deal, and we'll do the same down here. Looks like a little bit of an alligator, I guess. And my mind is going. Okay, good enough. Now let's continue with our endings here. If this is the 5' ending, just following it through, then this must be the 3' ending, right? And if this is the 3' of this strand, this must be the 5. And you can see how confusing it's going to get, because we have essentially the parental strains on the outside, and we're going to make the new daughter, if you want to think of it, or the new strands on the inside. Okay, so as you can see, we're going anti-parallel with these directionality of these carbons, and so will the new strand, a new copy. That's going to be happening here. We'll do that in different colors. Why not? I'm tall. So let's do that. Okay, let's go to magenta. Now, before we even do that, how are we going to do this? Well, we know that we unzip this. Helicase has done that. We know that there's some enzymes here, some stabilizing proteins that prevent, okay, these things from reconnecting again, because these could H-bond again, right? So we do have some proteins in here that are preventing this from going, stabilizing single-strand proteins. And I don't want to confuse things, so I'm kind of going to get rid of them. So we understand that they're there, preventing unzipping. Okay, now in order for us to create a new copy from this now template that's given to us by unzipping this DNA, we need a primer to start this. The DNA, as we just showed you, likes to connect to that third carbon, which has the hydroxyl group. The enzyme DNA polymerase, okay, can easily do that because of its form and function shape. Okay, not sure why that line was there, so let's get rid of that. Okay, so moving forward, let's continue with this madness. Let's move it over. What do we have here? Okay, so let's think about this for a second. We need a primer. Okay, so primase, and ending in ase, you'll hear a lot of these, is an enzyme that primes the DNA ending with, of course, a RNA linkage, a few RNAs linked. Now, RNA molecules are not double-stranded like this. They're not coiled up in a double helix. They're single-stranded, and we know that they are very similar to DNA, except for the uracil and, of course, the sugar is ribose. they degrade easily, and there's a lot of them in here, so it's a nice way to use. So DNA or primase actually will make a primer. So what we'll do is it'll prime, make a little strand and attach here. Now understand what's happening. Okay, this is the 5', this is the 3'. So if you think with me, this would be the 5' ending. And you can see we have some issues if I start this way. Now I wasn't sure, I'm just jumping head into this. What's the issue? If I prime this, DNA polymerase cannot build from a 5' ending. Say this has to be the 3' part of the primase, which could definitely do that. But the 5' ending is here. This is the 3', so this is the 5' ending of this DNA, of this RNA primer. And we can't add from the 5' ending, as we just talked about. So this won't work this way. So we're going to backtrack a little bit and say, well, that cannot be the leading strand. The leading strand likes to go toward the helicase in one motion. So this must be the leading strand. So primase comes, makes a couple of linkages, all right, and makes some single-stranded RNA, which is complementary to these bases here, and probably should write it like this. Here's the strand with its complementary bases, and notice this has to be the 5' ending, and now this is the 3' ending, and we add from the 3, right? Always add from the 3. So DNA polymerase, I'm just going to PLY, let's do 3. Okay, because of its form and function, its three-dimensional shape can add the nucleotides, okay, in this direction and basically move with the direction of helicase, which is moving this direction, unzipping the DNA. And I forgot to mention there's topoisomerase, which is another enzyme, which is actually cutting little pieces here to relieve the stress that the helicase is making by unwinding the DNA. Okay, so I forgot to mention topoisomerase is in work here doing the cutting a little bit so that the strain is relieved, because if it didn't, this would wind up like a plane and it would zip back on its head. So certainly very important there. So in any case, the leading strand is always identified by just the very easy directionality of the DNA polymerase going from a 3 to 5. And as fast as the helicase can unzip and topoisomerase can release the stress, this can move also. Now, you can see why this is the leading strand, because it goes in one direction, and of course there's no fragmentation going on, unlike the lagging strand that I have up here. Now, what makes this the lagging strand? Very simple, is that it can't go as fast as the leading because it's got to stop and start for obvious reasons. The first reason is I can't go back. Again, if I add a primer from primase, this is the 3', 5', I can't move in this direction. DNA polymerase is unable, because of its what? Three-dimensionality, do that. So we have to start sooner. So what happens is, very simple, is that once DNA exposes some new ground here, primase comes by and makes a fragment, a primer from RNA. And of course, if this is the 5', this is the 3' ending. And this is the 3' ending here. This is the 5' ending. We can't build from the 5th carbon on. So what happens is, by making a primer, okay, what happens is that the DNA polymerase can build from the 3 going this way. And of course, it will put DNA down. And as the helicase moves forward, you can see there's going to be more and more ground this way. And primase lays down another primer. Don't forget your orientation. Remember, this is the 5th carbon, so this must be the 3rd. And DNA polymerase can fill in that gap. So as helicase moves forward, it exposes more of the lagging strand and we keep going in that direction. Okazaki fragments, as you probably have learned, are these fragments from the primer to that new strand. All these are fragments because we can't go continuous like we go in the leading strand because we're always adding from the 3. We have to because we have to go from this position, so we have to wait for the helicase to go forward to expose more what? Unpaired nucleotides for making the new strand has to be given off or at least exposed. So we have to wait for that, and as we wait for that, we can add the primer, but always go back to the old fragment. So this is fragmented. It takes time because of the directionality of the DNA polymerase. Okay, and hope you can see that. Now, and that's essentially it. Now, of course, as we continue down this train, as we add the nucleotides and build this, okay, we know that the DNA polymerase is proofreading, okay, and pretty darn accurately too. I think it's 1 out of a billion errors are made in this process, which is pretty darn good, okay. And the problem is that we have a little bit of, we have an unconnected portion here, and we have a DNA, we have RNA here. So what we have is something called DNA polymerase 1 comes by, okay, removes the RNA and adds DNA. So any place where I have the RNA, any place where I have this RNA primer, DNA polymerase comes by, takes the RNA off, and you may say, well, why do you care if the RNA is there? Because RNA is not very stable. It does break down a lot easier than a DNA. Obviously, that's why we think that DNA is the hereditary information, as we've talked about evolution. So any case, the DNA polymerase 1 takes that down and adds the complementary nucleotides that you need to fit in those positions. And then, of course, ligase comes by and then connects those positions there. Okay, so that's how it moves. Now, one part I do want to make mention here is that when the helicase gets to the end, there's an interesting piece that we need to understand. So as we get to the end, so let's get to the end of this, and then we have this other strand, and we get, and of course we have our what? Our complementary pairing that occurred, DNA replication. So this was built, we removed all the RNA, but there's a problem here. And let's make sure we have our orientation here. This was the 5' end, this was the 3' end, and this is the 3' end, and this is the 5' end. If you noticed, why is there a little opening there? If I blow this up and make it bigger over here, here's my 3' and here's my 5. What happened was, to make this work, right, this strand here, if you notice, you always, every time helicase went forward and exposed some unpaired nucleotides, what went, RNA was used to code for, and I should write it this way. Just wait for me, wait for it. I should do it like this. So RNA made these linkages here. The rest of these linkages are regular DNA. The blue is just regular nucleotides that are base paired correctly. So this is the RNA. And we had to lay that down to make that, to go what? Always from the 3 to the 5. So we had to add from this direction, right? This is the 3' area. This is how we added. DNA polymerase can only add from that direction. So we were going in the same direction, just blowing up this area, the end of the chromosome or the end of the DNA here. The problem is when DNA polymerase 1, okay, takes away this RNA. Oops, not sure what just happened there. Oh, there we go, we're back. Once it takes it away, just didn't like that. This is now the 5' end. I can't add from the 5' end. because of what we just talked about. So what does that mean? Well, it means that this little piece of DNA, which, would have a, would be coded for right here, is now lost forever. You got rid of the RNA, and these base pairs have nothing to match with. We just lost a piece of the end of our DNA strand because we can't code. And remember, it's the 5' position. We cannot code for it back this way. So this is called a telomere, which I should say this is DNA shortening, and the area of the chromosome that builds in this problem is a telomere. A telomere is about, I think, 1,000 or 2,000 or so nucleotides long at the end of all the DNA strands in eukaryotes. And what it allows for, it builds in the idea that we're going to lose pieces of our chromosome, okay, over time. There's going to be chromosomal shortening at the ends. Every time there's a duplication, every time a new cell duplicates into the other, we're going to lose a little piece of that. In fact, people believe, and there's a strong evidence suggests that aging is due to this shortening of the DNA, because over time it's going to encroach on genes. Now, telomeres, interesting enough, are nucleotide codes, base pair codings are part of the, or what they are is they're part of the DNA that doesn't code for genes. So there's an ending part of every chromosome or every DNA fragment or that we're duplicating that doesn't code for genes. So it allows for this shortening. But over time, as we duplicate and replicate, I should say, our DNA, it shortens and shortens and shortens, and eventually it encroaches on parts of the DNA that actually codes for genes. And that's why eventually cells in cultures that they study eventually do not live after certain generations, and that's why they think people die of old age or have problems as they get older, because their cells have duplicated so many times to reach that age that they have now are starting to lose part of their genes. Okay, so that's really important, and there is some new technology showing that they can use something called telomerase to actually make this grow. Now, we do actually have in our bodies telomerase. Now, what do you think telomerase does? Well, telomerase is another enzyme, ends in ase, that actually increases this code. Not sure what's going on here. And it adds to the ending part. Okay, and telomerase is something that they're studying in large quantities in terms of trying to fight these aging disorders, especially ones where they think the genes that they have coded for are at the end. Okay, so in our bodies, we do make telomerase, and we use it for somatic cells, which makes a lot of sense, right? It would make a lot of sense for us to have somatic cells make sure that we're always adding to this ending. So telomerase adds nucleotides to the ending to make sure it's long enough so that when duplication occurs, and if you think about it, if you're making gametes that are eventually going to make a new zygote, you can understand why we want to make sure that ending is long enough so that our children, like being young, have plenty of time to lose all the nucleotides. If we were not to have telomerase happen in somatic cells or sex cells, then we would no longer have any fitness. Our youth wouldn't have fitness, and of course, the organism would not be able to continue on, and certainly the death of the species would occur. Okay, so understand it's pretty cool there. So there's a nice little picture where they have the telomeres at the end of the chromosomes. So very important part, and a lot of research happening in this area, as you can probably imagine. Hope that helped.